I am Adelodun Stephen Taiye from Babcock University, Nigeria. I am a faculty at the Department of Anatomy in the university. So I want to also welcome everyone to today's webinar, which is the second day of the webinar. And we are going to be looking at the light microscopy today. So the foundation has been laid for us yesterday by Olatunji, who has done justice to what the microscope is all about. So we are just going to be looking at one aspect of the microscope or microscopy, which is the light microscopy. This particular lecture is going to afford us the opportunity to be able to understand what are the principles and the mechanisms by which the light microscope works. Then it's also going to introduce us to how light interacts with matter so that we have better results in our imaging at the end of the day. So these are the learning goals for the lecture. At the end of the day, we should be able to describe what the light is and also explain the interaction of light with matter, like I said. Then we are going to describe what sample illumination is, in which polar illumination is one of them. Then we'll consider the bright field, the dark field, the polarized, and the stereomicroscopy. So these are all that we are going to be going through today. By way of introduction, as far back as the 11th century, people were using lenses as magnifiers. So they make use of lenses, place their object or whatever they want to view below the lens, and they discovered that the lenses were able to magnify the object or their specimen. But as time goes on, some few hundred years later, the first microscope was developed. That was after so many prototypes have been tested by various scientists, various inventors. So they came up at last with the first microscope, and that actually used a candle as a source of light. Advancement in technology now brought what we have today as the modern microscope. So the modern microscope, in essence, is nothing but just the fundamental basis or principle that is guiding the modern microscope is also what guides the first microscope that was invented. The only thing is that the modern microscope gave us the opportunity to be able to see many detailed things about what is happening around us. So we were able to look into our world properly, and it affords us the opportunity to be able to measure and diagnose some things around us. All right, talking about the light. Now, from our basic knowledge of physics, we all know that light is a component of the electromagnetic spectrum, alongside with others like the ultraviolet and the infrared rays. So light has its wavelengths within the range of 400 to 700 nanometer, and this visible light can be splitted or broken down into the various components of color spectrum or spectra. So that is what we can see at the bottom right of the screen. So light moves as a wave, and it carries its energy in form of what is known as a photon. So it is the interaction of this that brings about what we see under the microscope. All right, so everything that we see in our environment is just as a result of how the light interacts with matter. So when light interacts with matter, one of these processes will take place. So absorption can take place, reflection, refraction, diffraction, and the likes. Now, if I ask that, why do we see the tree being green in this picture all around us. And what happens to the other components of the electromagnetic waves of the light? The answer is very simple. Other components of the light electromagnetic waves have been absorbed, and it is only the green aspect that is actually reflected into the human eye. And that is why you see the leaf being green. Then the picture that we see at the bottom left of the screen also shows that the green ray is being absorbed, while we have the blue, the purple, and the orange being reflected. On the other hand, the picture at the bottom right is what helps to sum up all these interactions with light, with any matter. So the transparent parts will always allow the light to pass through unchanged, while the slightly opaque parts will absorb parts of the light, but also allow parts to pass through, but changing the amplitude, whereas the totally opaque part of it disallows any light from passing. So everything is absorbed, and the outcome is going to be total black. So the picture will be black at the end of the day. So that's what happens in total summary when light interacts with any matter. Now, let's look at refraction and diffraction as properties of light, putting the two of them side by side. Now, what is refraction? Now, when light, as it has been said yesterday by Olatunji, that light can move from one medium to another. So when light moves from a particular medium to another medium of different refractive indices, what happens is that the light can change its path or it will bend. Now, for example, in the case of the glass prism that we have, the visible light passes through the prism, and it bends in varying degrees. And it is this varying degrees that splits the white light into its 7 components of colors that we have. So we normally talk of red, the orange, the yellow color, the green, the blue, the indigo, and the violets. So it's as a result of refraction that this happens. The second example is what happens when you have a spoon inside a glass cup of water. Now, the spoon appears to be broken at the surface of the water. It is actually refraction that makes that to be. And coming to diffraction, when light moves or passes through a slit or a small hole, what it does is just to bend around that opening. And in this case, we call it the aperture. So it bends around the aperture. And an example of that is when light is shown on a CD or a DVD. At the back, we have the spaced track. So when it shines on it, it comes out, or let me say it is refracted, and the refraction looks like a rainbow, which all of us have observed in one time or the other. Now, coming to what is known as sample illumination. Sample illumination. There are 2 basic methods for sample illumination, and well, the first one is what is being applied to the older light microscopy. Now, they use what is known as the critical illumination, or what is also known as the Nelsonian illumination, named after the man who invented that. So what this does is that it focuses on an incandescent light on an image, but the image appears very bright. It appears very bright, but it is uneven. So this is one of the challenge that, or let me say the limitation that comes with the critical type of sample illumination. The second challenge of it is that the picture of the light source is also being captured in the resulting image at the end of the day. So there are two major issues with the critical sample illumination, and the next is what is known as the Köhler illumination. So thanks to August Köhler, who is actually a German scientist. Now, what Köhler did to solve the problem of the critical illumination method was just to adjust two components of the microscope. Now, in essence and in actual sense, the microscope, the mechanism of how the microscope works is all the components of the microscope are aligned on a single line, on a straight line, or let me say in a linear. So what the manufacturers just do is to collapse this linear arrangement of the components of the microscope and package it in such a way that it's going to be handy and it's going to look fanciful to us. But in the actual
Stephen, you are frozen. Can you just repeat that?
What Köhler did was just to
Stephen, perhaps if you switch off your own video, it might work. You are frozen on our side. Is he frozen on everyone else's as well?
Yeah, he's frozen for me too.
Okay. I didn't get you, Lizzie.
You were frozen for this last half. Maybe if you can switch off your video, the streaming from your computer will be better if you don't have your video on.
Okay, let me switch it off then.
Okay, and then from the Köhler elimination, if you can just restart that one, please.
Okay, from the previous slide?
No. We've heard that they put the components in a certain way, and I think it stopped basically where manufacturers put the particles in the line.
All right, thank you very much. Now, what I was trying to say is that the actual mechanism of how the microscope is arranged is in a linear plane, and what manufacturers just do is to collapse and package these linear arrangements of all the components of the microscope to make it look handy and fanciful for us to see. And that is what we carry all around. But Köhler was able to readjust, or let me say manipulate, the diaphragm and also the condenser so that he was now able to defocus the image of the light source in the plane of the specimen. So that is the breakthrough that we have against the critical type of illumination. And his efforts also gave us the opportunity to be able to eliminate some artifacts in imaging. So these are the two methods of sample illumination. Today, Köhler illumination procedure is very, very essential when talking about the principle upon which most of the light microscopes are being built, and even some other advanced microscopes, which include the confocal microscope, the DIC, and even the phase contrast microscope. So the Köhler illumination mechanism or form of sample illumination is very, very key. All right. My slide is not updating again. Hello, can anyone hear me?
We see the same slide now. Did you move on to a new slide?
Yeah, I'm trying to, but it's not updating any longer.
I can see your mouse moving, so it's not frozen.
All right, all right. I think it's there now, isn't it?
Yes, this slide's changed. Yes,
it has changed now. This is a demo. Hopefully, it will come out so that I want to now demonstrate, or let us watch what Köhler illumination procedure means. Now we are going to demonstrate that the essence of this is for us to align our microscope any moment or at any point in time that we want to do imaging. So it is a very important routine that every microscopist must always do so that you have a better result at the end of the day. All of us are very used to our cars. You know that when the alignment of your car is not in good shape, of course, the car goes any direction. But when it has a very good alignment, the car will be able to go on a straight path, as the case may be. So I'm going to play this so that you just follow and see what are the things we're supposed to do when it comes to polar illumination procedure before we start doing anything at all. So the number one step is for you to switch on your microscope, then you power it. Then the second step, you will bring in your
sorry, Sunday, you are frozen again.
interest.
Okay, you're going again.
Then after that, you adjust the diaphragm. You adjust the diaphragm to the extent that you almost have a black background, but you have a small view of the diaphragm. Then you adjust to sharpen the edge of the diaphragm. That's the next step. Then after that, you use the centering knob to centralize the diaphragm view. So you centralize it. Then after that, you open the diaphragm again to about 2/3 of the view that you want to actually look at. Then the next step is you remove one of the eyepieces. Then with your eye, you look deep into the eyepiece and now bring the edge of the diaphragm to the extent that you want it. Then you replace the eyepiece. Then after that, you are ready to go for your imaging. So I still want to recommend that these are something we can also help ourselves with, even on the YouTube. We can also watch it on the YouTube to have a better understanding of what the polar illumination procedure is all about. Like I said, it is always very essential that we do it in order for us to have a better contrast and a better imaging any moment or any time that we want to do photo micrography. All right, let's talk about the bright field microscope or microscopy. Now, the bright field uses transmitted light. Of course, the light is shown from below. Then you look or you view from the top, which majority of us are used to. So you have the light from below and you view from the top. Now, because the light bright field microscopy lacks, or let me say, it has a very low contrast. So it is very essential that we employ the use of dyes and stains in order to provide contrast for some of our biological samples. So that is very, very essential. And at the end of the day, the resulting image that you're going to see will be a dark object against a white or a bright background. A dark object against a bright background, and that is where the name comes from, bright field microscopy. So you view from the top, you have stains that are being employed for contrast, and this shows that it is not a so good method or technique for live cell imaging. So that is one of its limitations. All right, now how light travels in a bright field microscopy or microscope. Now, most of the time we use the halogen as a source of the light. So the halogen is turned on and it gives light, then which travels onto a mirror. The mirror collects the light and it sends it into a condenser. The condenser will focus the light onto our sample, which is on the stage of the microscope. Now, from the sample, the light that is transmitted enters into the objectives. Now, within the objective, there is magnification of this image, but not a complete magnification. So it goes on now into the eyepiece, and it is at the level of the eyepiece that the final magnification of the image is being established. However, sometimes we can fit a digital camera onto the microscope. So there are some that can fit a digital camera onto the microscope. And when this is done, there will be need for what is called a sensor to be able to capture the wavelengths. And the kind of sensor that we use is known as a photodetector. And one common example of photodetector is the closed couple detector. So it is in this chip that the ability to be able to capture the electromagnetic wave lies. Now, looking at the limitations of the bright field microscopy, a very thin sample is always needed. It's very, very needed. Now, that makes it very compulsory that sample preparation processes will have to take place. Well, there are still subsequent lectures we are going to listen to that will address sample preparation. So I want to enjoin everyone that we cannot afford to miss any part of this webinar series. So we are going to listen to the sample preparation. In that, we are going to appreciate what are the rigorous steps that are normally being taken in sample preparation. And so if one has to go through all these processes, you will know that it is not funny. But however, it is necessary when there is no any other option. Then another pitfall or limitation of bright field microscopy is that these processes of sample preparation can lead to the destruction of the specimen. So it can kill the specimen. And so live cell imaging is always very, very challenging and difficult with a bright field microscope. So to help that situation, one can decide to make use of other techniques like the DIC, the dark field microscope, and the phase contrast. All those can help in live cell imaging. Now, talking about the phase contrast microscopy. Now, it is an illumination technique that normally translates phase shift. It translates phase shift in the light that is passing through a sample to brightness changes in the image. So it makes use of what is known as phase shift, and the resulting image is enhanced so that the viewer will be able to see some details in an unstained or transparent specimen. So you'll be able to see some details that you will not naturally see under a bright field microscope. And when we are talking about phase shift in the phase contrast microscopy, all we are just talking about is the amplitude and the wavelength of the light. So in that case, a phase anomalous plate within that condenser changes the incoming phase of the light that is traveling to the sample, and this will create the constructive in-phase light. Then that phase anomalous is just a specialized device. It's a specialized condenser, and it's the one that creates that constructive changes. So then a corresponding phase plate within the objectives will now collect the light after the specimen. Then it blocks out the background and out of phase light. Now, interference is another property of the light. Now, when the wavelength or when light when wavelengths of light are in phase, or let me say they are aligned, what happens is that the amplitude that will result is going to be a constructive interference. So it's going to become a constructive interference, and that is what brings about bright imaging. So when lights are in phase and they are aligned, they will bring about a constructive interference, which brings about bright imaging. On the other hand, when light is not in phase, the rays of light are not in phase, then a destructive interference will result, and that is what brings about a dark image. Now, looking at the picture on the left side of the screen, it's the phase contrast microscope, which shows the position of the condenser annulus and also the position of the phase plates. Now, the image on the right side is the image of a bright field specimen and with its corresponding phase contrast image. Now, when you look at the two images, you will discover that the phase contrast image was able to reveal some details which we could not see in the phase in the bright phase microscopy image. So that is one of the importance of the phase contrast microscopy. It affords us the opportunity to be able to see some details that you cannot see even when a sample is not stained or it is transparent. So its advantages include excellent contrast of unstained specimens, and it can also be used with other imaging methods like fluorescence. However, it has its limitations as well, and some of its limitations is that it is not suitable for thick samples. It is not suitable for thick samples. Then the condenser annulus limits the numerical aperture. It limits the numerical aperture, and consequentially, it reduces the resolution power of that microscope. Then it also comes up with what is called an halo around the specimen edge. Permit me to go back to show that. So when you see the picture, which is the phase contrast picture, you will discover that the edges actually looks a bit blurred and it's not very sharp as well. So this is what halos is all about. Now, going to differential interference. Now, differential interference is another technique that can be used for live or unstained sample. It is also used for live sample imaging. And so the 3D contrast that is always being obtained is just by polarizing the light source, and this will create two planes of light by using a specialized kind of prism, and that prism is what is known as the Wallerstein prism. So the Wallerstein prism will split the ray of light into two, and the two of them will interact with the sample, and after that, a second prism will recombine those two planes and make it to become single. It is that single one that goes on into the eyepiece. So let's look at how this is achieved. We see the unpolarized light passing through a filter. Then the first prism, the Wallerstein prism, splits the ray of light into two. Now, these two will go on and on to interact with the sample. And after that, the second prism, which comes after the objective lens, will recombine the two planes again into just a single one. And that goes on into the eyepiece. Now, let's look at the dark field microscopy. The dark field microscopy it uses the scattering effects of light with matter to gather information around the sample. So that is its strength. It uses those lights that are scattered around the sample to gather information about that sample. So most of the incoming light into the sample are going to be blocked out, and it is only the light that is found around the edge, or let me say the outer rim of the object, that is going to be sent to that sample. So you always have a bright object against a dark background. So that is the reverse of what happens in the bright field microscopy. So in that field, you will have a bright object against a dark background. So this type of microscopy or techniques can be used to study or to look at biological fluids from animals and plants. Then in cell cultures, you can also use to look at microbes, food, and fibers. All right, now we have this picture that is showing the pathways for the dark field microscope, comparing it with that of the bright field. So the diagram shows the light paths to the sample in the bright field and in the dark field modes. And okay, sorry for that. So in the dark field, what helps or what is used to block out other lights apart from the ones that is coming from the edges or the scattered one is what is known as the patch spots. Yeah, so that is what is normally being used to stop or to block other lights. So unlike what happens in the bright field microscopy, so you have the central aperture where you block other lights. Then the second, sorry, so it is only the scattered lights that will now be used to image that object. Now, the polarized light microscopy, the light will be polarized in this situation by passing through a polarizing filter. Now, the filter has its own intricate property in which all the components or the property of the filter are in one orientation. So we only allow the oscillating light that is moving in a particular direction or in a particular plane to pass through, and any light which moves or travels in a plane that is not conforming to the plane of the filter will definitely be blocked out. So at the end of the day, there will be an analyzer that will now be used to bring those lights together. So you will use an analyzer to refract those lights from the sample and recombine them together later. So we make use of this polarized light microscopy to examine the optical property and the anisotropy property of matter. This particular picture is showing the image of a nylon taken by the light field, the bright field microscope, and also the polarized light microscopy. Now, we can actually note how the light is being refracted, causing the different colors that we can see in the polarized image. And also the background is showing that the only light that interacts with the object is all that is refracted. So it's a kind of techniques that can be used to study some materials like alloys, like metals, like ceramics and co. So like I said, the optical properties of matter and also its anisotropy can be studied or investigated by using these kind of techniques. So the last type of microscopy that we are going to talk about is the stereomicroscopy, which I also believe was mentioned yesterday. Now, all the other ones that we have talked about, all the other techniques we have talked about, all of them use either thin or transparent, very thin and transparent objects or matter. But in the case when you want to look into a more bulky and large samples, it is the stereomicroscope that is going to be the better choice to go for. So this normally it uses incident and reflected light to produce a magnified image of the sample. So it uses the principle of incident and reflected light to produce a magnified image. Now, one thing that is so unique about the stereomicroscope is that it has light source from the bottom, and it also has another one from the top. So the bottom light source is to help in case you want to look at some translucent material. So that gives a better view of those materials. And because we use it to dissect samples like invertebrate small organisms, the name is also known as dissecting microscope. Now, still on stereomicroscopy, the reflection. Now we know that when light shines on a very plain surface like a mirror, the angle at which light strikes the surface will be the same angle at which it reflects from that surface. So the angle of incidence is always equal to the angle of reflection when light strikes a smooth surface. But when it is a rough surface, then the angles will differ. The angle of incidence cannot be the same as the angle of reflection. In stereomicroscope, there are two separate light paths, and one is what is known as the Greenhoff design. And in this, each of the two eyepieces have its own objective in the pathway. The second one, okay, that is that. Then the second one is the Galilean system, and this allows light to travel through one big objective, and the light is refracted to form two parallel light paths with different optics in each. Now, in stereo microscopy, the viewer, when you look at the stereo microscope, the two eyes of the viewer always see the object differently, unlike in other microscopes. So the two eyes, they do not see the object as a single entity, and that is what brings about the 3D view of the sample that we normally see in a stereo microscope. All right, this is a slide of the spider, a stereo slide of the spider and that of the butterfly wings. And in modern stereo microscopy, it has a particular feature that allows someone to be able to take multiple focused images at different Z planes. So it's a feature that is so unique to a stereo microscopy. So it has that feature that allows you to be able to take multiple focused images at different Z planes. Now, these images can be stacked together to form a single image which is focused in all the planes. And this is a term that is known as the Z stacking in microscopy. So you have plenty of images that you can stack together, and all of them can be focused in all planes. So that is what is known as Z stacking. So finally, I want to leave this to everyone as a form of exercise, that we look at each of the picture and decide which of the techniques do you think has been used to capture each of these. I want to believe that you must have seen all of them, but as a form of exercise to ourselves, so we can take our time to decide which of the techniques has been used to actually capture each of these images. So, and that is the end of the presentation. I want to appreciate everyone that has taken his time to be part of the webinar for today. I say thank you to you all, and also I want to appreciate all the members of the ABIC family, especially everyone that has contributed one way or the other to the content of this particular lecture. And last but not the least, we are proud to be supported by the Charles Zuckerberg initiative. Without their help, this wouldn't have been made possible. So we say thank you unto them. Thank you very much, and God bless you.